Okay, so good afternoon and welcome everyone to today's Catalyze Accelerating Translation meeting. My name is Patrick Ahern with RTI and I'll be helping out with the virtual room today. So please note when you log in, your microphone and web camera will be automatically muted. But if you have any questions for our presenters, you can submit those into the Q&A window. Or you may also click the raise hand button and we may unmute your line to ask your question verbally. If you happen to run into any technical issues this afternoon, please reach out to me in the chat window and I can help troubleshoot. So at this time, Carmella, I'll turn the session over to you. Thank you.
Hi. Good afternoon, everyone. I'm so glad that you're here to listen to me talk for a little bit. I'm going to talk to you about CMC and how chemistry, manufacturing, and controls fits into the overall product development perspective, and give you some hints of things that I've learned over the years. Next slide. So we're gonna have three parts to the discussion, and after each one of the different sections, I'll stop for a few minutes if you want to ask me some questions. So I'll talk to you about some of the CMC points that I think are important for you to consider in early development. And then we're going to go on over to some specific information that for you to think about and to make sure that you have available to go into your manufacturing section of your application for the drug substance. And then we're going to go through a few things for drug product. Next slide. This is just a disclaimer. It's informational, but if you really have any questions, please give us a call or seek regulatory advice. Thanks. Next slide. Okay, so key points to consider in early development. Next slide. Okay, so from the FDA perspective, this is exactly what they say about the purpose of CMC. And it's basically that to assure that the drug sold to the public is safe and efficacious, as was determined in clinical studies, to make sure that the quality meets appropriate standards, and again, to make sure that the drug described in the prescribing information is actually what you're getting. Next slide. So this slide is the standard slide that you often see about how the chemistry manufacturing controls information fits into the overall clinical development phase. I have to tell you, there is no such thing as a 1-800-GET drug. There is a lot of work that's done behind that 1-800-GET drug that has to be done, and it's broken up mainly into drug substance or API, formulation, which is the drug product, and then the analytical development activities that go on during the development process. And so this just gives you an overall idea of what goes on. Next slide. So this slide gives you a little bit more information. It's actually for it's a biologics example, and it gives you a little bit more information, very similar to the other slide, but it gives you the time frame perspective relative to the overall development. And to be honest, I think this slide is fairly aggressive in how long it really takes to progress a biologics program, but this is what's out there. So next slide. Okay, so what are the critical elements of the chemistry, manufacturing, and controls elements of product development? And you have to consider how and where the drug is made, how the raw materials are tested and monitored, of course, the control procedures that are in place to make sure you get good quality and consistency, and that you have identified those quality attributes so that you can control them as the product development proceeds, that the test methods that you use to monitor product quality are accurate and appropriate, and also goes into stability. So what controls and how long does the product maintain its quality? Next slide. So again, I would love to be able to give you a checklist and say, okay, here are the standard things that need to be done in a development program, so that you can just perform them and check them off. But of course, that's not the case, that every product and every type of product, the manufacturing information is very specific to the product. Next slide. Okay, so what's really important too, and many of you are academic investigators or early small company investigators, and it's really important that at least through the development program, you have the right people on the team or the right expertise on the team at the right time. Okay, so it's not that everybody needs to be around the table at every project meeting, but you're eventually going to need people or expertise as noted in this circle. And from an overall perspective for early phase, many times these activities or functions are provided through the contract manufacturer. So if you're working with me on a program, oftentimes I'll say, okay, it's time now, I think, that we bring in the contract manufacturer because they can help us with specific aspects of the program. Next slide. Okay, so these are some key considerations, and I think they're relevant whether it's a small molecule or a large molecule biologic, or even a cell and gene therapy product. To me, your analytical methods and the analytical testing that you perform is basically your regulatory currency. And what I mean by that is that your analytical data buy you the good graces of regulators. And so the more analytical data you have, the more precise data that you have from your analytical methods, it gets you through the regulatory process much more easily. And so you're going to need analytical testing and test methods for monitoring in process while you manufacture for the drug substance and the drug product, and for actual drug product release. One of the things that you're going to hear are the acronyms or phrases that if you go to CMC meetings in the recent past, you're going to hear critical quality attributes. And basically, the critical quality attributes are those attributes that when you change them, you change the quality of the product. And so you need to identify those critical quality attributes using a well-qualified method early on. And again, those critical quality attributes are the test that you will generally put a specification or a limit on. Next slide. Okay, the other thing that is important to consider is the control of your raw materials. And I think this is especially true for biologics and gene therapy programs, and also for programs where you may be developing a device for implant, it's really important that you start with pharmaceutical grade material as early as you can. And the reason I say that is because no matter what anybody tells you or the specifications that they have for a raw material, oftentimes there are components or things about that raw material that will change. And especially for critical components, it will end up causing you issues with your program if there are slight differences in the raw materials that you choose. So I recommend that you go early on to pharmaceutical manufacturers and get CMC or pharmaceutical CGMP or pharmaceutical grade raw materials early on to use in your processes. And some of the information that you need, even at early phase IND, you're going to need to set some specifications with the vendor, or at least review the specifications and make sure they're relevant and appropriate for your product. Information that's now required for raw materials, especially anything that's of biological origin, you need a BSc or a TSE certification to go along with your certificate of analysis. And again, especially for biologics programs, you will likely want to perform use tests. And what I mean by that is define the basic parameters that the raw material needs to meet to be able to function appropriately in your manufacturing process. For early phase, it's usually not time effective to identify and qualify multiple suppliers. Certainly, if you can, that's a good thing. But by the time you get further down in your development program, you are absolutely going to want to qualify multiple suppliers of your key raw materials. You want to think out for the long haul, think out, whether your supplier is going to be able to supply your raw material for the next 20 years. Next slide. And so all of this information goes actually into your manufacturing process. And again, for biologics and gene therapies especially, one of the things that you want to consider is that when you're developing your methods, your manufacturing process, you need to think about whether the tests that you're performing or the process that you're performing is readily scalable to large scale. So oftentimes, you will get investigators that have used solvents, for example, like toluene or something like that, use in small quantities isn't really a problem. But you have to think about the fact that you're not going to be able to use large quantities of solvents like toluene as you scale up. So you really want to develop your manufacturing processes where you have laboratory and raw materials and things that can be scaled. And again, what you're going to want to do is, as soon as you can, you're going to want to define those critical process parameters. And then you're going to have to put acceptable ranges to those process parameters as you go along in development. And the key thing to remember is that FDA does expect the specifications and the testing that you perform on your manufactured product to change and to tighten as you go through the development process. So keep in mind when you are setting your specifications that you don't want to make them too tight in the early process. It is much easier to tighten a specification from a regulatory perspective than it is to loosen a specification. And then again, another term that you're going to hear oftentimes is quality by design. And basically that concept is, instead of trying to consider that your analytical testing is all you need to show quality, that really what you need to do is you need to understand what your critical quality attributes are and develop your manufacturing processes using quality by design principles. Next slide. Okay, I put this slide in here about drug master files, and drug master files are important in many development programs, and like especially for small molecules where you may be purchasing your active ingredient from another country. Then most of the time the information that for the manufacturer of the active ingredient or the drug substance is going to be provided to the regulators in a drug master file. Okay, remember, or things to remember about a drug master file is that the information is submitted to the agency, but the FDA does not review a drug master file for quality and acceptability until your application comes in and you cross-reference that drug master file. And they will then look at that information in that drug master file relative to how that material fits into your manufacturing process. So don't just assume that because you get a drug master file number from a vendor that means anything about acceptability, because it doesn't. You're gonna wanna make sure that you have an agreement with the whoever is providing you with the drug master file information. Make sure you have an agreement that if there is an issue that the FDA finds with the drug master file, that you have some sort of time limit or something in your quality master agreement that says that they have to respond quickly and efficiently to all of the issues that are raised. Because one of the other things is that when the FDA reviews the drug master file relative to your application, if they find deficiencies in the drug master file, they will send the holder of the drug master file the listing of the specific deficiencies that were found. However, the letter that the group that is cross-referencing the drug master file gets from the FDA is just that there were deficiencies found. It does not tell you what those specific deficiencies are. So you need to have an agreement, again, with your the holder of the drug master file that they will provide you with regulatory feedback when and if they get feedback from the agency. Okay. The other thing is that it is possible also for you to prepare a drug master file and submit information in a drug master file. It's rarely done. That information is generally included in your own CMC section, but it is possible to submit information to the agency in a drug master file. Next slide. Okay, so quality assurance. Of course, you heard in our last presentation from Diane Earp. She is our quality guru here at RTI, and you will hear her talk about quality assurance and how important it is. But it is very important for the release of the drug substance and the drug product batches, for review and approval of your standard operating procedures, also release of raw materials, and review of batch records and deviations, and putting into place any corrective action, preventative action plans, and validation protocols. The one important thing to remember is that FDA does not require that you have a specific separate quality assurance group at early phase, okay, for like GMP manufacture. It's understood that when you have very few people in your company, that multiple people are going to need to wear multiple hats. And so you will want to have someone designated as your quality assurance person. And that, again, just like for recommended for GMPs, you're going to want that person to have the capability to actually say, no, this doesn't meet the quality that we need, and make decisions that's different from the manufacturing folks. We're going to talk more about reference standards later, so we'll go on. Next slide. Okay, and so again, you hear all the time about CGMPs. What that is current good manufacturing practices. Those are the goals that are necessary to ensure the same or product is the quality it needs to be. It's the processes and the checks and balances that you have in place to make sure that you can make the product consistently and that it will have the appropriate quality attributes throughout the shelf life. So the chemistry, the review of the CMC information and compliance to current good manufacturing practices, they're not the same, but they do very much overlap. And at marketing approval application, one of the things that you're going to have is a GMP audit as a part of the review of your marketing application. And the other thing that I want to bring out is the C. C stands for current. And what that means specifically is that the agency expects that the good manufacturing practices that you're using are current relative to state-of-the-art information. Okay, so you cannot, if like now, if you wanted to run a procedure that was standard 15, 20 years ago, and there's another procedure that's being used routinely and is more accurate and more suitable, then the FDA expects you to modify and make sure that your procedures are current relative to state of the art. Next slide. And as I was saying earlier, the FDA does expect that the extent of the good manufacturing practices that are applied during development does change, they increase, and what you have to have from early phase to later phase changes and progresses. And this is just an example of some of the things and how the current good manufacturing practices change and increase during your development program. Next slide. Okay, so this is a stop point, and wanted to check to see if there are any questions about the information I've provided so far.
Carmella, I do see one question. Will the slides be made available?
The slides are going to be made available. Yes, they will be on the Catalyze website when it's up and running. But if my information is on the last slide, if you want to send me an email, I'll send you a copy of the slides too, if you'd like. Anything else? Okay, we're going to go on now. Okay, so now I'm going to talk about some of the drug substance information that is included in a regulatory submission. And one of the things that we try to do is to make sure during a development program that you're focused, and this is the whole what NHLBI is also trying to stress, is that the work that you're doing is translatable to a product, okay? And so therefore, one of the things that we have to consider in development is that you're always asking the question, you're always going to need to ask a question about your program. Is this a must-have or is this a nice-to-have? Because, you know, when you're doing normally in research, you perform every study that you think that you need to perform to answer any question. But in development, not saying that you shouldn't perform all the studies that you need, but you're going to need to understand and consider acceptable risk. The other thing is, like I have here on CTD format, I don't know if many of you have filed INDs in the past, but the traditional format for the IND is has, I think it's 9 different sections to the IND. But when FDA made the requirement that INDs for commercial activities or commercial suppliers needs to be electronic, they implemented the requirement that all of the submissions need to be in the common technical dossier format. So again, there's a very, and what that CTD format is, a very prescribed sections that are needed for completion for each one, from the CMC, from the clinical, from the non-clinical perspective. And so what I'm showing you here is actually some of the major headings in Module 3, which is the quality section or the CMC section for the Common Technical Dossier. And these are some of the major section headings that are included in the CTD. I'm going to go into detail about a few of these sections, but that doesn't mean, because I'm not covering everything, that doesn't mean that each and every one of them isn't as important. It's just so that I want to bring out some important points in these sections. And we can, if you want to contact me later, I can work through you and help answer some of the other questions about the CTD format, and also help you understand what information goes in there. Next slide. Okay, so the description of the manufacturing process and the process controls. Okay, one of the main things that you're going to need, and I always ask for when I'm talking to sponsors and with programs, is the FDA wants your manufacturing process, they want it to be in a flow chart. They want to be able to see the overall manufacturing process visually. And then they want you to include a narrative description of each one of the manufacturing steps. So they use the process flow chart and the narrative description to make sure they understand the overall manufacturing process. And in the process description, you may provide, that's where you can provide a bit more information. But another point to remember about your manufacturing process information that goes in your IND, you do not need to be specific to equipment or even necessarily to specific ranges like stirring speeds or stirring times, those sorts of things. So you can provide a very general flow chart with a bit more information in the narrative, but it does not need to be so specific that you have to actually include ranges. Now, as your manufacturing process gets more mature and as you go further in your clinical program, then you are going to start needing to add ranges, specific ranges to your processing parameters. But you don't need to do that at the very start of your program. And if you are going to include a range or process range or something, don't make it too tight. Okay. And again, what you're going to do is through that manufacturing process, you're going to find out which of these steps or which of these components makes up the critical quality attributes that you need to be concerned about. Next slide. Okay, the other thing that's really important to understand is impurities. So from the FDA perspective and other regulatory agency perspective, an impurity is anything that is not your product, not your active ingredient. And that's the same, it's true for a biologic or a gene therapy product. If there are some flexibilities with biologics that if you have your compound is slightly different, say in structure, but it still has equivalent activity, then that's not considered an impurity. But anything else that's in there that is not an excipient is an impurity. So there are multiple types of impurities. There are inorganic impurities like catalysts, reagents, ligands, salts, charcoal, other things. Those are considered impurities. Organic impurities, which are anything that comes in maybe from your starting material or is brought in through the manufacturing process. Organic impurities are degradation products. So you need to understand that. And then, of course, residual solvents are considered impurities. So you have to control those to appropriate levels. And the other thing that we don't have on here is elemental impurities. Okay, so elemental impurities are really important to understand and control. And you're going to need to provide test procedures and limits for those impurities in your manufacturing process, in your regulatory documentation. And again, from the start, you don't want those to be so tight that it's impossible for you to meet those specifications. But what you need to understand is that the agency is going to expect you to tighten those specifications as your development program progresses. Another thing that's important to remember is, and I will tell people that I'm working with, sometimes you want your batches, that you're going to use in your IND enabling toxicology studies. Sometimes you want to consider having those batches to be a little dirtier than what you would use for your GMP processes, because the FDA expects that any impurity that is in your product has also been covered or was present in the toxicology studies that you performed. And so what happens is that during scale up sometimes, or specifically if you change the starting material or you change your synthetic process, what happens is you get different impurities. And what you're going to need to do when that happens is you're going to need to go to your non-clinical, your toxicology expert and say, look, we have an impurity that's showing up. It comes up all the time. We can't really get it out. And then they're going to have to consider whether you're going to need to do a bridging toxicology study to get that tox coverage for that impurity. Next slide. And specifications. So again, specifications are really those tests with limits that you propose to show that you have appropriate batch control and quality parameters for your product from early phase through commercial manufacture and beyond. And those, you have to include acceptable limits for each of those tests. Usually the components or the things that you have determined to be the critical quality attributes in your manufacturing process are often the components or the parts of the process where you will actually put a specification limit. Again, early phase to later phase, don't make them too tight to start with. FDA understands that you have to have some batch history and that it takes that batch history to be able to understand and how to tighten your process and be able to achieve those limits. So your specifications will tighten as you go along in your process. You'll always want to make sure that you have a certificate of analysis or batch analysis for your batches that are used in your clinical and non-clinical program. And you have to include that batch analysis data in your regulatory submissions. Okay, and so then the testing that you normally apply for, say, release and those specification limits, those ranges, are often the exact same things that you're going to test on stability. And you're going to need to meet those ranges on stability throughout the entire life of the product. And while, again, you start early, you do not have to have your methods validated at the early phase manufacturing process. Usually what FDA has in guidance documents is you have to have your validation of data or your information on validation of your method available at phase 2. I would tell you differently. I would say validate or qualify your methods as early on as you can, because again, your information is only as good as your method. Next slide. And this is just a slide for a small molecule that shows you some of the information, the tests, and some of the specification limits that are included. And I'm not going to go into internal testing right now, but for sure, those are the specifications that you need at release and on stability. Next slide. Okay, so again, my favorite part of all of this is analytical methods, again, because I think your analytical methods are so very important in your overall development program, because again, what quality and information that you know about your manufacturing process is only as good as your analytical methods are. And so your analytical procedures are there for ID, quality, purity, strength, okay? And what you have to do is you have to include a description of your analytical method in your regulatory submission. Again, at early phase, you don't have to provide as much information about your method as later phase, but you do have to provide information about your analytical test methods. And the other thing that's really important is it's a bit harder for folks that are doing gene therapy or biologics manufacture, because oftentimes their methods have to be very specialized for your process. But we always check to see, look in the USP and F, okay, and use USP and F methods when possible. So use those basic methods, because again, what's important about that is that from the FDA perspective, they consider that those USP NF methods are essentially validated. And so the only thing that you have to do is qualify those methods as being appropriate for your process. And again, you don't necessarily need, from a validation perspective, precision and robustness of your method until phase 3. Next slide. Okay, this is the part, justification of specifications. Boy, I get lots of grumbling of justification of specifications, but basically, even at early phase, FDA expects, or regulatory agencies in general, expect you to be able to explain and justify why you're doing the testing that you're doing and why you're putting the ranges that you're putting on your manufacturing process. And again, most of the time, you know, your information comes from your batch production and process knowledge. You know, that's how you justify. But what you're going to want to do is you want to make sure that you can justify your specifications based on product characteristics and process capabilities. Next slide. Okay, reference standards or materials. You always, for an HPLC method, like an HPLC method is considered an indirect method by the FDA. And what that means is that you're always comparing the data from one batch to your reference standard. So it's always a comparison. And a direct method would be considered like NMR or IR, that actually is measured by the instrument, a specific spectrum relative to the active ingredient. So it's always important that you have qualified your reference standard early on that you're going to use. And for gene therapy and biologics programs, you always have to compare back to those reference standards. So those are absolutely gold. Okay, they are the what you have to show that your product is the same from the most important perspectives, from the time you're doing your toxicology studies throughout the time that you submit your marketing application, and then through commercial supply. So if I could put in your mind anything at all, reference standards are very important. Make sure that you set aside enough reference standard under the most stable conditions so that you will have that material to bridge back to. There are, during the development program, you're going to need reference standards for potency assay, for your metabolites, your impurities, your degradation products. But again, you don't have to have all of that information at phase 1. And also too, depending on what your active is, you may be able to purchase a reference standard from the USP. Next slide. Okay. So that is a breakpoint for the drug substance part of our discussion. Are there any questions?
There is one, Carmella. It says, this question regards when to onboard SME in a biotech startup developing a biologic. Can you discuss at what stage, for example, pre-CDMO selection, pre-GLP batch, or pre-GMP batch, would you recommend to have the SME present?
And just make sure I understand the acronym. Is that the subject matter expert? Okay, I'm going to talk to, okay, I'm going to talk about the subject matter expert. From my perspective, that person is needed at the very start, way before you ever even get to candidate selection, because there's a lot of information that they can provide and help you think about what's important in developing your product. And your subject matter expert also is, we're not going to talk about this today, but is also important in helping you from the very start develop your targeted product profile and also your quality target product profile. So again, I think they need to be involved very early on. Next, any more questions?
So there is a part B to that.
Okay.
So which subject matter expert, for example, analytical methods versus CMC versus formulation or regulatory, and at which level, as an FTE or as a consultant, should the subject matter expert be present?
Yeah, I think what's important is that you're going to need your experts in the specific areas, usually at different times in your overall development program. Okay, and the amount of time that you're going to need them is going to change in your overall development program also. So early on, again, if it's a small molecule or a biologic or something like that, then you're going to need your person that understands. You're going to need the person that is helping you to develop your active ingredient. Uh-oh. I'm sorry, guys, my slides just went out. Can you still hear me?
We can hear you, Carmella.
Okay. I'm gonna push down here on another screen. Just one second. I'm sorry about this. Okay, so again, you're going to need that person early on in your process to help you define your process, help you develop your master cell bank or your working cell bank and qualify that. And then you're also going to need your analytical person early on to help you develop your methods. But then again, you're not going to need your formulation person probably at the very start at candidate selection. You're probably going to need that person a little bit later on. So it's a situation of when the expertise moves into the project team and moves out of the project team. Does that make sense? Okay, so now I'm going to go on to the drug product information for, again, it's the next slide shows you. Are you guys still there?
Yes, we're still here, Carmella.
Okay, so so it's still going to be the information that we're providing is in the common technical dossier format. And again, I'm going to cover specific information, not every section. Next slide. Okay. All right, so again, we're going to talk about the manufacturing process and process controls. Again, what you're going to want, same thing as with the drug substance. You need to have a process flow chart and you need a narrative to explain the process controls and what you're trying to do. And it's very important if there are any intermediates or isolated intermediates or anything that are involved in your process, you make sure that that's clearly understood and identified. Next slide. Okay, so you're going to want to controls of critical steps and intermediates. That's what I was saying before, is that those critical steps, again, are the steps that you're going to apply, most likely a test and a specification. And you need to consider the inputs to your manufacturing process as well as the outputs to your manufacturing process, and make sure that that's controlled. And that, again, if you have any isolated intermediates during your manufacturing process, for example, I'll explain what I mean about an intermediate. We have a project where there is we're spray drying the active ingredient. Okay, and so that's a part of the drug product manufacture. And if you spray dry that, your active ingredient, then you're going to want, that's going to be an isolated intermediate. You're going to want to do some testing on that. But then when that active ingredient goes into your final product with your excipients and things, then that's the actual final product. Next slide. Again, like we were saying, for raw materials in general, you're going to want to make sure that you have you've selected your excipients or your inactive ingredients that go into your formulation, and that there are the appropriate quality that you're going to need, and that they're sourced from suppliers that are going to be able to supply you for the long haul. Then you're gonna want to make sure that when possible that they're manufactured under good manufacturing practices. And again, from a biologics perspective or for a device manufacturing perspective, it's a good idea, when possible, not to use novel excipients And the reason I say that is because there is this handy dandy book called the inactive ingredient list, or this list called the inactive ingredient list. And on that list, it provides the inactive ingredients that have been used in products of the same type, for example, an oral or an injectable or whatever. And when you're using those excipients that have already been used in another product of the same type at the concentration or the amount that you're using or below, then you do not have to do further qualification, i.e. further toxicology studies or anything like that on your inactive ingredient. Next slide. Again, you're going to have specifications for your drug products, just like you do for your drug substance. You're going to make one of the main things that you're going to need to understand, even for tablets. Now, the FDA expects you to have microbial enumeration test performed on your finished products, your drug products. And of course, if it's an injectable product, that it's sterile. But many of the tests and things that you're going to need are similar to what's in the drug substance, but you're going to need drug product specific methods like dissolution. And again, what you're going to want to do is go to the USP/NF first for your method, for method descriptions, and use the USP/NF methods when possible. Next slide. Okay, so stability summary and conclusions. I didn't say this for the drug substance, but you have to have stability testing performed on your active ingredient from the date of manufacture until the point in time when that active ingredient is incorporated into your drug product. Okay, so if you're going to use your active ingredient, you know, in a couple of months' time and incorporate it into your drug product, then you don't need to have, at early phase, you don't need to have 36 months of data. You only need enough data to cover from the date of manufacture until it's incorporated into the drug product. For the drug product, again, most of the time, the testing that you're going to do encompasses what your specifications mainly are. You're going to want to make sure that you're using the right conditions. And usually, you know, you think of it as a temperature with a relative humidity, but keep in mind that an increased relative humidity, meaning more moisture, is not necessarily the worst case scenario for your product. Sometimes it's going to be better to test at that temperature with lower humidity than a higher humidity. So you really need to think about what are the conditions that are appropriate, or what are the most challenging conditions for your product. Again, the FDA does not expect you, again, to use necessarily the exact ICH conditions for your stability studies at early phase. And the other thing is that, again, what you need to support your IND is you need stability data on your clinical trial material that supports from the date of manufacture until the point in time that that drug product is used in your clinical study. Okay, so if you have a short clinical study and you go from manufacture to the clinical study ending in 6 months, then all the stability that you have to have is 6 months stability. What's really important, though, is that you will, if when possible, you will want to start your stability study a month, 2 months, if possible, before you actually go into your clinical study, so that you have, if your product does go out of stability, that you have enough time to remove that product from the clinical study. Because that's exactly what happens. If your product goes outside of your proposed specifications in your clinical study, then you need to remove it from the clinical study. And that's the commitment you need to make in your regulatory application. Another thing that's important is that in the United States, you do not have to have an expiration date on your drug product label, because again, the FDA understands that usually you'll put a 6-month use date on your clinical trial material, but it's understood that close to that 6-month date, you're going to do a retest. If it's still stable, you're going to extend the expiration dating period. So that is why you do not have to have a an expiration date on your clinical trial material. Okay, that's all for me. Thank you very much for listening. Do we have any questions on the last part of the presentation?
Carmella, I do not see any questions for the last part.
Okay. I've included some helpful links on the next slide, as well as my contact information in case you want to ask some questions, or Kim indicated that the slides would be available, but